Welcome if you just joined us online. Okay, I was just talking about this. So if you joined from Sheffield, you know all this anyway. So I was just saying about the two groups that I belong to, explaining where is Sheffield and our history with the steel industry, but also the lovely countryside around to persuade people to come and visit. So one of the things Sheffield was quite famous for was material science and the metals industry and the development of stainless steel. And for the biologists, I should mention that we also had Hans Krebs comes from Sheffield, and one of our research buildings that a lot of the biomaterials and tissue engineering research takes place in is called the Croteau Research Institute, and that was named from Harry Croteau, who got a chemistry prize. So you see, I can satisfy all the disciplines with our famous scientists and engineers that came from Sheffield. And I just thought this was a nice thing to mention. Just literally 4 days ago, I was at this award ceremony in one of our Cutler's Halls, which is one of the historical buildings from this trade, the metals trade, where famous metals companies contribute to this organisation. And they give awards to students. And this day, our student Lukasz happens to be Polish, but that's not related to the story. And this British student, James, they announced his award and then said he's going to come to Krakow and work with Sano. So Sano was mentioned at this prestigious event, which was really nice. I don't know if anyone knows who's hosting James, but he's coming this summer, working on something way beyond my understanding about blood flow and red blood cells and flow. So that was really nice to think that our historical materials institutes are still involved in this very modern discipline of what's going on in Sano. So there's some nice further connections there. So what is Insignio? So Insignio has a remit very much like Sano. It's about healthcare and using engineering, biological, chemistry and computational methods to support healthcare questions. So it's a very large institute across the faculties and with several hospitals. And the branch that I belong to is this branch here, biomechanics, biomaterials and cell engineering. So we partner with Sano. So just to show here, we're feeding in to the Sano ecosystem, and a lot of you, I guess, belong to different parts of Sano here on this diagram. And we do a lot of work. So we're just mentioning with Thomas Elliott with digital twin work and all the different areas that we have computational methods and digital twin work involved. So this, if you're interested in any of these, come to me and I'll tell you which colleagues to contact. So one of the new things we've just started is women's health technology. So we noticed there was quite a gap and little research focusing directly on problems of women's health, which are often neglected and thought as not as significant. And I'll give you a more specific example here, these meshes that I'm showing. But if anyone is particularly working on anything to do with placenta, reproduction, fertility, or menopause, then we've got a lot of researchers just starting in that field looking for collaborations. So here's some examples here. So Shannon, or Shannon from our group, has a model of placenta and looking at blood flow in placenta, and she looks at cervix and devices that were used for people having problems keeping pregnancies, and there's a whole group working in that area with some very interesting models, which again, don't ask me about the models, just ask me about the contacts, and they shall tell you all about their models. So I showed you those plastic meshes in that previous slide. So these meshes, I don't say in Poland, but they caused a big scandal in the UK because they were used for hernias quite well, but when they started being used in women for pelvic organ prolapse and stress incontinence, they caused terrible problems in the reproductive organs, tearings and infections, and it was seen they were just basically the wrong mechanical properties. They weren't right for that region, and that hadn't been properly tested or thought through or modelled. So we have people who look at retrieval of the implants and looking where the damage is, and people that are doing manufacturing of more improved materials that could be used in pelvic floor more safely and avoid these terrible problems that happened. So how about myself? So how did I end up in Sheffield? I sort of did a full circle and ended up back where I started. So I studied originally in Manchester and York, which are two cities very near Sheffield. Then I moved to the ETH in Zurich, where I worked on bone structure. And I did some time in the United States, in Hershey, Pennsylvania, also in orthopedics, in Philadelphia in dental materials, and then in Chicago when the field of tissue engineering was becoming very popular. And that's where I learned a bit more about combining the cells and materials to create new tissue structures. And then I moved to Sheffield to start my faculty position. So here in Philadelphia is where I met Anna Isichka, my best friend from Krakow, and the reason I visit here quite regularly. So hope Anna's listening online. And it's really nice to be expanding our network of people between Sheffield and Krakow. So finally, we have a building named after a woman in Sheffield. So Pam Liversidge, who is one of the cutlers of that important organisation I told you about, and our building is named after her. And this is our laboratory. So anyone who is an experimentalist and might want to visit, we have all facilities for bone cell culture, bone analysis, sectioning bone, imaging, high-resolution imaging, and so on. So why do we work in the musculoskeletal area? So for this, I wanted to first start with my colleague's work, Tekla Bonsi, who works on whole body biomechanics. So talking about the multi-scale aspect. As she points out, mobility is really the sixth vital sign of somebody's health. If you're not mobile, then firstly, it's a sign that something's probably wrong with your neurological system. You have other health conditions that are impairing your mobility. And there is a feedback loop where as soon as your mobility is affected, you're more likely to develop a whole load of conditions we know about, like cardiovascular conditions and so on. So understanding people's mobility abilities and trying to improve it is very important. And in her particular case, she works on different sensors and measuring systems and monitoring systems that can be used to test, for example, if an intervention is working, or to understand why a condition is having so many effects. So we need to look at mobility loss. So then within our group, we basically work in all the different aspects of the musculoskeletal system. So here's an example here from Chen on modelling brain damage. So basically looking at the skull and then the corresponding damage on the internal tissues after trauma. He looks at things like helmet design to protect brain trauma. He looks at responses to the brain after a stroke or different kinds of damage. He does go on to use like AI tools and those kind of modelling to try and understand the images, compare images and see what that means in terms of the brain damage that might be caused. And then here's an example of one of our digital twin projects that two of my colleagues are involved in with the spine. So the spine is one of the most important musculoskeletal organs, I would argue, because it's keeping us upright. And when the spine is damaged, again, neuromuscular system is closely linked, which means you have terrible pain, terrible mobility problems, and lower back pain is the biggest cause of loss of earnings for most working age people. So we have projects on spine metastases, so showing the gaps in the spine, spinal repair, and whether spinal fusion is right for a patient, so patient-specific models of spinal fusion to try and predict whether that's a good idea. And then also in the field, close to my field of biomaterials and tissue engineering, we have a lot of projects on ways to maybe for the nucleus pulposus of the disc, or spine, cartilage bone co-culture implants, and ways to understand the spine better with explant experimental models. And this is combined with the computer modelling of the other colleagues to fully understand the spine in a holistic way. And then from the spine, we go to the limbs. So we have work in both upper and lower limbs. So here's some upper limb examples from my colleague Jen, trying to understand grip and movement of the arm, which is obviously very important, again, for overall health, diseases that affect the shoulders, and diseases that affect dexterity. And so trying to model, image and model what's happening in these situations. We have Pinaki's work on mostly hips and other kind of limb implants, looking at implant loosening with models. So we've got the metal and the bone separately modelled here, trying to look at virtual populations to then assess, what's the likelihood of success of certain implant technologies, and he looks at osteoporosis and side falls and how that affects hip failure. And then we go all the way down to the ankle, where my colleague Claire is particularly interested in osteoarthritis of the ankle, because this is a less studied joint, but very important, especially in sports. So it happens to young people. Sporting injuries can cause ankle damage that then lasts for the rest of your life. And again, she does a combination of modelling, biomaterial solutions, imaging, and we have a project at the moment on testing whole ankles, donated whole ankles from deceased donors, where we're trying to understand the complexity of the joint movements. It's a very complex joint, and that's why there's not really any suitable implants created for the ankle yet. And then one of Dana's projects is on arthritis from the basic modelling point of view, all the inputs, the inflammation, the fat tissue, the signalling pathways that are involved, and trying to better model the sort of multi-condition inputs that cause osteoarthritis and the cell biology behind it. Okay, so that's an overview of the group as a whole and the different things that are going on in Sheffield. And then for the second half of talk, I'll just talk about my own research, which is very experimental, but I'll give you a few little hints where I think modelling could really enhance the work we're doing here. So I'll be presenting work from these various members of my group, particularly these more recent students. So Ilona, Reilly, and there's Caitlin here somewhere, and Caitlin. And these are some of our funders. So particularly we've got good funding on spine research from Spinner from the EU, and here's Sano here. So in my group specifically, but you'll see already overlap with some of the things I told you about, we work on mechanobiology, so understanding how cells themselves respond to some of the forces I was telling you about that occur at the whole body level. We work on the biomaterials that are used for orthopedics and how to improve their chemistry, structure, integration with the body. And we work on the biomechanics of the tissue, so how to improve its strength. For example, if we're engineering a bone tissue, it needs to mineralize well so that it will be able to function inside the body. So what can we do to improve its matrix and its mineralization? Here's some micro CT scan of a tissue engineered structure that was loaded using principles of mechanobiology, which then improved the amount of mineral that was present there. So these three topics are integrated to create better bone, basically. So I mean, we mainly look at two scales of the bone structure. The scale where our cells live, so the osteocyte, the cuni, and the scale of the materials composite scale, which is collagen fibers with mineral crystals inside them. So, and this is a nice picture for the neuroscientists of osteocytes, and they're canalically connected inside the bone, because people often don't realise it's just as complex as a neural network and just as interesting. So if anyone wants to make a network model of osteocytes and they're canalically, I think this could be a really fascinating area. And as I was just telling Thomas, it seems like bacteria go right inside there as well. So now you can model the cells, the bone, the canalically, and the bacteria coming in. Right, so this was where I first got into this field. So I first got into this field trying to understand how and why bone responds to loading and how it fractures. So using horses, because race horses actually have very strong bones, as you might think. So when a horse is racing for, money, basically, not the horse gets any of the money, but anyway, the bones are strengthened, they're toughened, particularly, probably more important than the strength, their ability to resist damage and cracking. So their work to failure is the thing that really improves the more they run. And this is similar to humans, where basically we put on bone mass if we exercise our bones. So tennis players' arms are a really good example. And I did quite a lot of work looking at the orientation of the bone structure and how that is very dependent on the load in our body. So where we have bones that are under compression a lot, they will reorientate to resist compression. And where we're stretching them a lot, they'll reorientate to resist stretch. So it is an ongoing live process that the bone's adapting to the loading within its region. So it's something we're all familiar with from the gym, that our muscles are adapting to the load, but you don't always realise that the bone is as well. So I'm going to be talking about creating new bone from stem cells. So I'll just explain which stem cells will be coming up. So we work mostly with mesenchymal stem cells. So they're the ones that come really from the bone marrow. And we also have a model of bone differentiation that's called NLOA5. That's a highly mineralizing osteoblast. So some of the work is with those. But we can get mesenchymal stem cells from many sources for therapeutic use or to make model systems, including adipose tissue. And I think Anna's got some good work in that, if anyone wants to talk to her about that. From the periosteum, which is the skin that surrounds the bone. Umbilical cord blood is a well-known good source, and so on. So the cells I'm interested in, that they can differentiate to, are these osteocytes and a little bit on chondrocytes, but other people use them for these other kinds of tissues. So in particular, I'm going to be using, in fact, for the work I'm telling today, probably mostly this cell line. So this is a cell line developed at the University of York by Paul Jennifer. And basically this just demonstrates it can convert to bone, it can make collagen and calcium, which is always, so the inputs we're always looking for is, can it make collagen and calcium? Because this means it can make the two main components of the bone matrix. So when I came to Sheffield, my idea was to move to 3D, because bone, as you saw, is a really complex 3D environment, and a lot of bone cell biology had been done on monolayer culture and flat culture. So the first material I used for this was just a simple polyurethane foam. So this is just a commercial foam that's used, you might be sitting on it under there. It's used to cushion lots of things. It's used to insulate pipes. But it's a really nice open-celled foam that's extremely resilient to regular loading, which is why it is a quite useful biomaterial. So what we find is if we grow cells on this foam, it will fill up the spaces, it will lay down matrix, and we can load it and see a response. So we see more calcium on loading. I showed you an example of that earlier. So this is the kind of work that I've been doing even before Sano was invented, with colleagues from Krakow. So a colleague came to me, a student came on Erasmus and used this foam to look at fluid flow. If we don't actually load the whole foam, but we perfuse, do we get similar results? And we do, and she found, there's no time to go into all the detail, but she found some really interesting, very specific effects depending exactly when she turns on the perfusion, when she turns it off, how many times. So the cells are very sensitive to a mechanical regimen, and you have to really tailor that to get the result you want. Hence why it's very difficult to do enough experiments to get the exact result you want, especially with flow responses. It's, there's so many confounding complementary variables. So modelling of these flow responses would be a very interesting project, I think, instead of us just empirically checking all these different time points and lengths of time of loading. So one thing we need to think about when we think about growing cells in 3D is the fact that the cells I showed you, they were really embedded in bone matrix and rounded up. They weren't flat on a surface like they are in the foams I showed you. And this, of course, will strongly affect their mechanical situation, right? Because the shape, what they're attached to, that's going to affect what kind of biology they do. So, so one challenge is really to find or create materials that are a bit more representative of the body, that that might house the cells in a bit more of a realistic environment. So that's what we're doing here. So my colleague, who's a chemist, Fred Claessens, has lots of different polymer processing methods. But one of the ones we like very much is high internal phase emulsion. So that's when you have two immiscible fluids, you make an emulsion like mayonnaise. He's Belgian, so he loves to use mayonnaise as the example. But unlike mayonnaise, you don't keep it as a paste, you polymerize it until it's completely solid and wash out the internal phase. So that gives you this bubble structure where all the bubbles are touching. So you have all these windows. So you have large pores and then interconnecting pores. And depending what polymer you make it of, you can make a really quite stiff structure or a very flexible structure. So this is really useful for mimicking tissue properties and microenvironments. So we can use it to make beads, discs. We can 3D print it, use the paste as a something for 3D printing, and I'll show you some examples for that in a while. And as we know, 3D printing can be based on medical scans. That's like a big advance of 3D printing, that you can take a scan and then convert it to a replica. And this is basically an experimental digital twin in a way, can be used, it's used even by surgeons to practice surgery and so on. So we can replicate trabecular bone structure, for example, from a CT scan provided in this case by Enrico. So this just shows you what this looks like zoomed in. So we can see that we have the printed 3D structure, but internal pores that are not printed but created by the fact that the paste was a height, the ink was a height. So we can get this multiscale porosity that's on the order of magnitude of bone scales, because bone scales are 100 to 500 for the bone marrow spaces and only about 10 or 20 for the cell spaces. So we can see cells grow on this really nicely because they can infiltrate the both layers of porosity. And they can differentiate to osteocyte-like cells. So I showed you that osteocytes connect. They live in lacunae and then connect, and we can see this histologically and in the SEM that we are, okay, not as pretty as those lovely canaliculi I showed you, but certainly able to do canalicular-like connections. So when we look at these heights, we've got two applications really, and I'll show you a couple of examples. Could be used clinically, so it could be an improved implant material, or it could be used as a model system to understand the bone and the signalling of the bone cells better. So here's one clinical application, because a recently finished project by a student who was looking at periosteum, so the skin that wraps around the bone. In a large bone trauma, the surgeon would like to peel off some periosteum from a healthy site and put it as a sheet over the repairing site, because it helps the bone regenerate much faster. But this means you have to lose periosteum from a healthy bone, and this is kind of a vital part of the bone that provides stem cells and so on. So could we create an artificial periosteum that would have the same abilities? So this is the idea here. So using the hype, so this hype was made of a combination of our two favourite polymers, polycaprolactone, that's quite a stiff one, and polyglycerol sebosate, that's quite a soft, flexible one. The sebosate is too soft, the caprolactone is too stiff, so she made a blend and got really ideal properties between the two, something that's easy to handle and wrap around, but still has this multiscale porosity. And then the idea is to encourage angiogenesis. That's one of the main benefits of periosteum, that blood vessel invasion comes. So anyone who works on modelling blood vessel invasion, that's something we're very interested in the tissue engineering field. So we can use a sugar, deoxy-D-ribose, encourages blood vessel invasion. And that can be seen here on the CAM assay. So this is a really nice experimental model assay as well, if anyone that models this kind of thing. Easier to access than a full animal model. You just break the egg, you put the material on the surface of the egg once the shell has been removed, and then because the egg's growing, it continues to make blood vessels, and they can infiltrate the material. You can count how many are coming and their size and diameter and everything. So we have done work with a mathematician in Oxford who models this kind of behaviour. So we can look at the number of blood vessels. We can see that our sugar is improving that, and therefore making more periosteal-like. And then we can use an ex vivo bone defect from the chicken. So again, because this is such an early stage chicken, it's not an animal experiment. It's before the time when it would be classified as a full animal. And the sample can be wrapped around it, and we can look at bone growth across the defect, which is mimicking a broken bone. Okay, so another project collaboration with Krakow was a visit of Claudia Slowick, who came on a Marie Curie-funded staff exchanges project and further developed these scaffolds, this time for a multi-tissue model. So here she was looking at a bone layer and a gingiva layer to mimic the oral cavity and the alveolar bone. And here's some genetics for the genetics people. So basically all these different genes are expressed at different amounts depending, again, what the material is. So the material is very important for directing stem cell growth, and you always need to optimize the best chemistry and porosity of the material to get the tissues you're interested in. So again, I won't go through all these in detail. If anyone's interested, we can talk about it afterwards. But we eventually do get a nice model of the alveolar, the fibrous tissue, and the oral mucosa layer that can be used either for testing. So here they want to test drug diffusion through that model. So if a drug was delivered just with an oral patch rather than having to swallow it, how would it go through the system? And then to model defects here, like a dry socket, like when a tooth is removed and you end up with an empty space and it often gets infected. Okay, but in the beginning, I told you that mechanobiology was important, and I showed you my polyurethane foam, and I said that we loaded that and got different results. So with this polyhype structure, it would be good if we could also add mechanical loading. So that's what we're now doing here. So we tried to create a device that was a little bit like a microfluidic device, but larger, enough scope to actually grow a 3D bone culture. And then this would be a way to interrogate how cells are behaving in a dynamic environment. So basically, using that height, we made the structure, then we flowed through with these syringes. So we're flowing through the microfluidic devices. Those are embedded with differentiating bone cells. And in this case, we're looking at a particular mechanobiology structure, which is a cilia, a primary cilia on the cells. So this cilia, with this drug, will be activate the system, and therefore we can read out the activation, and therefore this model system is a good way to test the drugs that are involved in bone formation and mechanobiology. And it looked like it was, because with the drug plus flow, we got the best results for differentiation. Very important area is cancer invasion, you might know. So cancer invasion into bone, that's a secondary cancer that's very common and very difficult to treat. So here we're using the same system, this HYPE system, to look at how white blood cells might support or protect from cancer invasion. So first we looked at the effect of white blood cells going into bone and how flow affects that, and whether the cells, I don't know if anyone's got an immunology background, but if you don't, these might not mean much to you, but the M1, M2 phenotypes, which are the more active, more protective types of white blood cells, and how they behave differently in the system. And then we add the cancer cells. So now we've got cancer cells being added on top, the white blood cells either activated or not, and the bone, because we know that there's immune modulation of how those cancer cells will invade. And again, I won't have time to go through all this, but you see we've got differences. So basically some cancer cells invade faster than others, and we could replicate that in the system. And some white blood cells have a stronger protective effect than others, and we could replicate that too. So that's the combination of the bone cell, the two types of white blood cell, and then the two types of cancer cells invading. And we see a change in depth of invasion in our different systems. So again, that's quite complex to set up, needs a lot of optimization, and there's only so many scenarios you can model, as in experimentally model. This is the kind of thing that would be really conducive to computer modeling to look for, to compare scenarios, predictive scenarios. So Damien Lacroix, one of my colleagues, used to do, and still is working a little bit in this area of modeling what's happening in these kind of flow systems and scaffolds. So he found, for example, that a lot of the scaffold designs, the actual output isn't really how the design looks, especially with 3D printing, and that can strongly affect the flows and the mechanical forces inside that scaffold. So first that has to be better optimized. And then he looked at where cells are likely to land, depending on the flow and the scaffold design, and then where the tissue is likely to form, and trying to optimize the scaffold shape for the kind of tissue that you're trying to create. So the last story really is about that second level of microstructure I told you about, the lamella level. So these bone lamellae are really important. So anyone that's come from a composites background, you know that when you have a fiber composite, the fibers affect crack movement and propagation, and that's going to affect the overall toughness of your structure. That's how bones don't break more than they do. And this is something that's often neglected in all the systems I told you. They didn't have any lamellae or fiber structure. They just had some extracellular matrix. So how could we mimic this better, and what could we then do with that? So here we use a fabrication technique called electrospinning. So here you spin thin fibers of polymer onto sheets. So that's just a foil sheet collecting. And this is what they look like. They just look like very thin whitish sheets, which are made of polymer fibers, which you can collect so that they just align randomly and kind of fall in a higgledy-piggledy shape, or you can spin while you're collecting and pull your fibers into a straight line. So when we do this, I don't know if this will work as a video. Yes, it will. We can get aligned fibers. So that's the green lines there. No, sorry, that was the red lines, which we've lost. The green lines are the collagen fibers made by the cells. So what we see is that, I think I'll run it again so I can talk through it. The red fibers are the scaffold. The green fibers are the collagen, which aligns with the scaffold because it's being guided by these fibers here. But then as we grow and build collagen too far away from the scaffold, it stops aligning. It made that kind of more of a rounded honeycomb shape. So that guidance by those biomaterial fibers is helping the structure form more like a bone structure. So we wanted to know if a stem cell could actually make lamellae in the lab based on that system. So we used, again, polycaprolactone, aligned versus non-aligned, and grew stem cells on it, and let them kind of build up collagen, and then image it using second harmonic generation imaging, which can image collagen without staining. So you can image it over time and over quite a depth, like hundreds of microns of depth. So what you see here is this nice alignment, but we're already seeing a change in orientation as we come away from the original fibers. And if we measure this, we can see a peak of orientation which shifts as we go through the samples. And if we didn't see the cells on aligned fibers, if it was just one of those other scaffolds I showed you, that wouldn't have any orientation. So it really is the biomaterial that's allowing this orientation to occur. And then the twisting, however, is not something we created, it's something the cells or the collagen, we're not sure which, is doing spontaneously based on the previous layer. So that's very interesting, but why do we care? Well, we care because there are diseases where this doesn't happen correctly. So diseases of collagen mutations, where the collagen doesn't align properly, the bone structure is not strong and tough, and there's brittle bones. Bones fail easily. So this happens, it can be the collagen 1 or the collagen 2 pro-collagen that assembles the fibers, and then other associated genes as well can have the same outcome. And there's really huge variety in the severity of this disease. So it's not yet quite well understood. It's understood that any mutation is not great, but some mutations, very mild outcome, where some cause a really severe change in the bone quality. And you can't always predict, especially when new ones come along, you can't always predict how severe that will be. So a model system where we could grow the matrix in the lab could be a way to understand this better. So the idea is that when we use these aligned fibers, we can straight away see the difference between a healthy and an osteogenesis imperfecta cell. So here we've collected only fibroblasts from the skin, because it's difficult to collect bone from somebody with a bone disease, as you might imagine. So, but the fibroblast alignment does happen on these scaffolds as well. And we can see healthy cells aligning nicely, or patient cells, although they do align, much less, more of a spread. And then if we don't have aligned scaffolds, we don't get this alignment. Okay, so the idea is that we can, we're also doing it with antibody staining when we don't have access to SHG, and that works fine as well. So we're doing this, again, we're seeing osteogenesis imperfecta much more disorganized, much less clear, strong alignment and dense matrix. And the idea is to take this forward by mutating the cells. So difficult to get patient cells, as I said, and it's difficult to make them bone-like, but we can take iPS cells, so we can take fibroblasts and reprogram them to iPS, and one of our colleagues is doing that. Or we could try a CRISPR knockout technique where we mutate the cells and knock out the particular genes that we think are the most important, and look at the outcome in terms of, do we now replicate this kind of structure compared to this one? Okay, so just a couple of slides now, in case any ceramics people made it. So I did mention bone has mineral in, but all the scaffolds I told you about were polymers based. So if we think about bone hydroxyapatite, is that something we can work on? Can we incorporate that in our scaffolds to make implants that are more biomimetic? And can we modify the hydroxyapatite to have important properties like angiogenesis for the blood flow that I told you about was important, or antibacterial to resist implant infection and so on? So yes, we can. So both the systems I told you, both the electrospun and the 3D printed, we can mix hydroxyapatite into our solutions in various concentrations, and we can get similar structures, but these would have like nano hydroxyapatite crystals embedded in them. And that trabecular bone I showed you actually did have that system, so it had hydroxyapatite embedded in the matrix. And then back to some modelling, so that I can finish with some modelling for you and something to think about. So a new postdoc who's just come to my group, funded by the Royal Society of Britain, works on really the spine structure of the hydroxyapatite, so the crystal structure. So it's quite a simple, relatively calcium phosphate structure. but it can have vacancies and it can have substitutions. And this is what can change its properties quite dramatically. Some very interesting substitutions for medical applications are zinc, copper, and strontium. So these are things we're going to start looking at with his work and how that might affect the crystal properties, which affect their mechanical and electrical properties, and therefore their how they support cells. So, in conclusion, I would say that 3D scaffolds better represent the structure and mechanics of bone tissue. So that should lead to more relevant culture environments, both for a bone implant and for a bone model. And we need to collaborate more. This is the most important conclusion. Everyone should come to Sheffield from Krakow. Everyone from Sheffield wants to come and visit your lovely city. And we've been having relationships for 25 years, me and Anna, might even be more like 30. Now we've got all these new relationships for the last, is it 13 years of Sano now? Or how long has Sano been going to it? 10?
6 years.
Only 6? Seems like it was longer. So we're going to build on this, carry on our collaborations and our good relationships, and hopefully create some fantastic healthcare projects for Europeans' population health. Okay, thank you very much.
Thank you. I hope you can answer some questions if there are any.
Don't ask questions about the models. I'll tell you who to talk to.
So to folks online, maybe let's start with,
should I
have any questions from the room?
You can ask very basic questions because I know it was a very mixed talk. So, yeah.
Yeah, so I'm curious about like infections if these implants were made into the patients. Like what affects that? Is it like vascularity or what kind of properties of the material? Like
that affects whether it gets an infection. Yeah, well, as usual with these things, the most important thing is the doctor. So the infection route is usually bacteria from the skin that accidentally get inserted with the implant. Sometimes, if the company is not sterilised well, the implant itself might carry an infection, but that's pretty rare. What happens is infection gets introduced during surgery. But if it's introduced onto the implant, that's what's really dangerous, because then the implant goes into the person with some bacteria, starts creating biofilms, and then you have a really difficult to treat infection because it's coating the artificial material and interacting with the cells that are trying to ingrow. Does vascularization affect it? That's an excellent question. I know we definitely have a problem of when there's a lack of vascularization in bone, we get necrotic areas, and that then becomes infected. So I know that way around it's very important. So lack of vascularization leads to infection. Whether vascularization improvement can also clear the infection, I'm not sure, but that likely would be, because usually as soon as you improve vascularization, you sort of improve white blood cell access, and I'm not sure if that's been looked at though. But I imagine there's definitely probably a feedback loop relationship between vascularization, biofilm, and progression of the infection. Yeah.
Any other questions? Actually,
well, how long can you use this implant? Because I've heard from some family stories that in Poland, for example, when you break your hip and you are an older person, you are more likely to get an implant which is cheaper, but you can use it only like 10 years. And sometimes there are young people who break the hip and where they are very unlucky to get the implant for 10 years. So I don't know.
That's exactly true. So I think the best ones last 20 years. So you're right.
Yeah.
So they usually say 15 is kind of, I think it's something like 85% of implants should last 15 years for it to be like an approved implant. There's some kind of regulation along those lines.
And the size there is also in
yeah. So partly it's the actual metal. So titanium being a better metal than stainless steel, but more expensive. The coating, so the hydroxyapatite coating will make it last longer, but that's more expensive, and so on. So a solid permanent implant, yes, should last at least 15 years, maybe longer. And so someone that has one young often has 3 in their life. If you have your first one at 40 because it was a sporting injury or trauma, you might well have 2 or 3 in your whole life. And every time it's revised, it's worse, because every time it's revised, you're digging away more bone and you have more empty space that needs filling. So that's that type of implant. The other type of implant, the ones that are designed for bone to grow around them, at the moment, a lot of those are permanent. They're these kind of metal meshes I was showing you, or sometimes they're hydroxyapatite granules. That's the one we're trying to replace with biodegradable materials. Because the idea is if we could have a biodegradable bone graft, where the bone would gradually grow back and replace. The hope is those might only be needed for, let's say, 6 months or a year, and gradually the bone fills in. But they'll probably never replace the full joint implant because they're not strong enough and large enough. So in terms of the hip implant, all you can do is improve the bonding to the bone to try and make it last longer.
General question
Yes.
about the time durability.
Exactly. So yeah, at the moment, I don't think anyone's improved on 20 years. I think individuals might last longer than 20 years, but you wouldn't expect it to last longer than 20 years, really.
Yeah, thanks for the answer.
I guess there is still a lot to discover, but you showcased some, technological advancements, both coming from like the biology side, looking at stem cells and trying to understand this front, and then more from this kind of technological side in 3D printing. Like, where do you see the future going, or like, do you see this kind of being the two solutions where we actually find those future applications?
Yeah. So I think in orthopedics, it's probably more the 3D printing is for clinical use, because the stem cells, they're a great system to understand, and they're fascinating cells in terms of helping us understand development, but they clinically, there's so many problems to overcome in regulating stem cells and not knowing what the outcomes will be after long-term use, and could they have cancer risk and so on. I think in the case of orthopedics, where it's not life critical, probably stem cell use will be quite minimal. Where we think they might be used is, for example, things that are secreted by stem cells, like the extracellular vesicles, or we're looking at using stem cell matrix as a natural implant, if you like. But I think probably not clinical use of stem cells. So there, I think the 3D printing is really the way forward, because if you can print a smart scaffold with the clues that we know the stem cells respond to, like the correct porosity and mechanical properties, then hopefully you'll get stem cell invasion from the body's bone marrow, and you wouldn't need to implant the cells. That's all. So understanding how the stem cells work with the material is important, but clinically, I think making that material as smart as possible will be the direction. Does that answer your question?
Yeah, that's fascinating as well.
Well, thanks, interesting talk. Are there any databases in scaffold John wanted by? No, but there should be. Good proposal. Sell it to the EU. No, if you talk to Damien Lacroix, that's the kind of thing we started talking about. And I've seen a few people proposing this kind of thing, but there's no really good systematic analysis of that. There's lots of papers where they may have varied 2 or 3 issues with the geometry with one particular material, and then a little bit then estimated the amount of matrix production, but there's no comprehensive analysis. And I think this is really limiting our field, because everyone just tries empirically, whatever their printer can do or whatever their method can do. Test 2 or 3 porosities, which is the most you can manage in biological testing because of the time it takes, and we don't have a good database. No. So please create one. I'll be very happy.
Great, but that sounds like a really fantastic opportunity for more computational people.
Yeah, exactly. Oh, that would definitely need some AI and whatever, because of the complexity of the geometry, especially if we are going to take into account curvature and porosity and numbers of pores, and there's so many things to optimize. Yeah.
Yes,
Oh, yep.
Question about some computational requirements. Do you see some challenges in all these applications about size of the computing resources required, or maybe some complexity of the workflow?
I can just say big. I don't understand enough how you estimate how much computer size you need, but I do imagine it's huge. Especially if again, it depends how many interventions you want to test with your model, right? But if you are going to test the range of interventions that are possible in scaffold design, you would need a really big, complex model, because there are so many variables. Because if on top of all the scaffold porosity, design, shape of the pores, size of the pores, and so on, you're then going to look at the flow through each of those different designs, and then how strong the flow is, how many times, how.
So you can look at it as a kind of virtual population.
Exactly. Yeah. So I think it would be
virtual patients or.
Yeah. So it can be done 2 ways. It can be done really using trabecular bone scans of patients, but then you would need a nano CT scan if you can look at the internal porosity of the canaliculi and so on, or by scaffold design and think of it as something that you're just going to put patient cells on and then implant, and then you've got slightly simpler geometry, slightly, but because of the range of possibilities, that's still extremely computationally heavy. And then if you are going to model separately the polymer and the mineral, at another level of complexity, those things I told you about, the mineral crystal structure inside the scaffold, and what happens if I add zinc, what happens if I don't, and how does that change all my outcomes? That would be, yeah, big job.
But that some computational chemistry is inevitable.
Yes, yeah. So what we're trying to do in our group is sort of multi-scale models where we have one at the, let's say, the chemical scale, looking at the hydroxyapatite crystal, and the input of that feeds into the next sort of metascale model and so on, because you're not going to be able to do it all in one model. So it's more like model systems that then give you boundary conditions for the next model system, I guess. Hope I'm not saying that wrong if Enrico's still there.
Do you have any more questions?
Maybe thank you for the talk. So you mentioned that clinically preparing like scaffolding with cells inside from the patient is difficult.
Yeah.
I wonder, how do you actually put the stem cells into the biomaterial, say the bone implant, and if you put them inside the patient, do they have any chance of like surviving and doing anything?
Okay, that's a great question. So the one thing that is already done, certainly in trials, if not currently, is just take just the bone marrow. Don't even try and separate the cells or sort them or anything. You just take some bone marrow, mix it with some media and put it into your scaffold. So I've seen that done. That would be fairly simple to do, but you don't really know the biological outcome of that. But because if it's the patient's own cells, likely they will survive, but is it going to help at all? It's a bit difficult to know. I've had some surgeons say they think it does. The next more complex thing would be, prior to an operation, extract cells, purify, get the stem cells, help them differentiate, and return them. And then you would have to sort of inject them into your scaffold, or use a really modern method of 3D printing where the cells are in the ink. So the bio-ink method, if you have that. So you might have a special polymer gel that's very mild, that's not toxic. Usually it's made with gelatin or some kind of polyethylene glycol, and you would print your cells inside the matrix and then implant that. But that's, as a clinical technique, that's going to be really tricky. The patient would really need that structure, because you take the cells before, print it, but then it would need to be implanted quite quickly after printing to keep the cells on. So other methods are just kind of injection. So one thing we're trying to look at is injectable materials. So I didn't really have time. I only showed you one slide of when we make a bead, but those beads are pretty small, just a few hundred microns. They are injectable. So you can make a paste that had beads of your scaffold or micro particles of your scaffold that might be injectable into something like a spine defect. So that's a situation where that might be a way, and you could mix that with cells fairly easily then.
Is any of this available clinically, or is it like purely experimental?
Some of the materials are. So, for example, ceramic. Some of the ceramic materials as a paste or putty or granules, definitely that's commercially available. Some of those polymers are used for other types of applications like sutures and things like that. I don't think there's a polymer composite on the market that I know of at the moment, apart from with natural. So, for example, there's hydroxyapatite collagen sponges available on the market, that kind of thing. The small defects, like small defects in the knee and things like that, that's sold by a company we collaborated with in Italy called Ceramica. So those are available. All sorts of kind of demineralized bone is available, and collagen, fabricated collagen structures. As far as I know, nothing with stem cells clinically, officially anyway, clinically used. And a lot of those composites are not yet commercially available.
Thank you.
Folks, just on the organizational front, I know we're running over time, so being mindful of other possible commitments, please, feel free to leave.
Yeah.
But since we've got more questions, let's keep the discussion going.
Main challenges of integrating in vitro models within silico. Well, I mean, I think it's just what you said in the first place, the computational time. And well, there's two things. The usual thing with computational models, garbage in, garbage out problem. So it's do we yet have enough inputs? Because it's quite difficult to measure a lot of these things. And certainly every lab working on mechanobiology is measuring something differently. So it's even working out what is the significant input the model should work with that we can collect from the model systems we do use. and then try and make the simulation, as I said, reproduce the complexity of it. Like when we know there's, we get a different result if we stimulate for 5 minutes or an hour. We get a different result if we stimulate an hour every day to an hour once every 5 days. We get all of these things. So how, I guess we need to understand the mechanism of that first, so that the model can simulate that mechanism. Otherwise, the model's just flailing around. I guess unless we have some kind of stochastic model that does look at all the statistical possibilities. So yes, there's a lot of challenges. So making sure we don't put garbage in, and then the number of different scenarios that need to be modelled. That makes sense?
Great. We'll go to the next question.
Mm-hmm.
No more questions?
Like we are done now.
I think that's one more.
Oh, one more.
There we go.
I'm just hypothesizing.
So
we said that stem cells themselves are problematic. So maybe let's take a step back, put some kind of bio-friendly material like bone scaffolders or something to encourage the stem cells from the bone present or other tissues around to invade the material.
Yes.
But could we put some, I don't know, peptides or some chemicals to help them? Invite them to
that is definitely a direction that I think is more promising and that quite a few people are working in. So yes, originally people were working on proteins that we know stimulate bone growth, like a bone morphogenetic protein. In fact, that's commercially available. Was a scaffold with, was that calcium phosphate? I think with bone morphogenetic protein in it. And that is quite expensive. And definitely, if Anna's still there, have to be careful. So one of Anna's favourite proteins, bone morphogenetic protein. So she's doing a lot more interesting work on it and how to use it. And it works better in certain conditions. So it's kind of trying to optimise those conditions. But it is super expensive for for regular use, like across the globe. And it can stimulate too much bone. So there was a problem where actually the company was sued because there was bone overgrowth. And so if bone overgrows like around the neck, which it was doing, it then presses on the nerves, and then people are having terrible pain and what called osteophytes, like spicules of bone sticking out of their vertebrae and things like that. So you have to be careful with proteins that stimulate anything, as we know. So the angiogenesis, for sure, people are looking at VEGF-loaded scaffolds or chemicals that stimulate VEGF to stimulate angiogenesis, but then you've got to be careful of cancer. So if you have something that overstimulates angiogenesis, you're going to stimulate tumor angiogenesis. So basically, as we know, any biological molecule is going to have side effects, and we've always got to weigh up, is it worth it so that's why we rather go the chemistry route. So I think we're more interested in modifying the hydroxyapatite, because as I said, some of those are angiogenic, some of those stimulate stem cells. And maybe I think using sort of pure chemistry rather than these complex proteins that affect all different signaling pathways might be a safer way to go. But people are trying the whole range.
Thank you.
Great.
I also have one question. So before the clinical implication of the in vitro studies, do you do any in vivo studies? And if so, do you meet any challenges or disappointment if there's something like work in vitro but in vivo in animal? If you do so, there's oh no, that's not how it's supposed to work. So let's go back in vitro and try it.
Yeah. So personally, I don't do much, but it's typical animal study people do with these kind of things. Is one's called a calvareal defect. So you cut a hole in the skull, put your material, does the bone grow fast over it? Or you might do a little leg like the one I showed you on the chicken, but you might do that in a rat or something that's a little bit bigger. In fact, that's the BMP story, is a bit like that. So BMP worked really effectively in all the animal models, and then in humans was much more variable, the response, because our animals are just carefully genetically controlled, aren't they? And then as soon as you get to humans who aren't carefully genetically controlled, which is a good thing, you get much more variability. That's one of the problems. Second problem when it comes to bone specifically is our biomechanics are so different. So if there's anything that is involving a combination with biomechanics, the loading system is so variable. We do have work, so Enrico does a lot of work on mouse models where he loads the mouse bone and looks at drugs and looks at the combined effects. And there definitely is some value in having loaded bone plus, for example, PTH was a drug he looked at. It's a hormone that stimulates bone. But translating that to humans is very hard, because how do you make a human do the equivalent of the exercise of when he was loading a mouse for so many cycles at exactly a certain time of the day and so on. So one thing, I guess, the people doing whole body analysis need to look at is what kind of exercise and physiotherapy produces the local loads that are beneficial. But even that is not very well known. We know jumping's good, for example, but what that's doing at the bone strain at the local level is still not very clear. But certain exercise definitely more beneficial than others, and you can't really reproduce those in animals very well. And then the problem of age, because really the biggest problem in bone is when we're older, and it's very difficult to age an animal, so expensive, takes a long time. We have postmenopausal effects in women, which to mimic in an animal, we have to cut the ovaries off. So that's a bit of an artificial menopause. So lots of things it's difficult to mimic in the animal, which is why we'd like to have in vitro models with human cells, which we think will hopefully give us more humanized response, and then go to computer models that will give us a more humanized response, hopefully. Oh, there's something else in the queue now. That was the same thing, wasn't it? We haven't missed anything.
Oh yeah, I think you spoke.
Oops. Oh, that was Anna. Oh, listening here more on the in vitro, but I think we sort of said that. No, we haven't vascularized in vitro yet. The only vascularization we've done is putting it in the chicken, because that is a really cool area. And one lady in Galway is doing some, Leisha McManara, is doing some vascularization using endothelial cells in a similar model and making little networks, but we so far haven't done that.
I don't see any more questions.
Okay.
Very lively discussion. So thank you again, Gwen.
Thank you everyone for coming.